I am Anita Rathi, Associate Professor Botany, Government College Girls, Gurugram. Under the aegis of Director General Higher Education Haryana, I'll take the topic of nostoc, its structure, reproduction, and importance. This topic is in continuation with my previous topic, that was cyanophycy. So today we will talk about nostoc, an example of cyanobacteria. The genus of nostoc, it includes about 29 species. Mostly are in colonial forms. They grow in forms of mucilaginous balls. So we'll take the taxonomic position first. Kingdom is bacteria. As you all know, it is cyanobacteria. Phylum Cyanobacteria. Class Cyanophyceae or Myxophyceae. Order Nostocales. Family Nostocaceae. Genus Nostoc. So we'll talk about the occurrence of nostoc. It basically occurs in freshwater ponds. There are large colonies which are free floating. You can see circular balls there. They can be attached or they can be submerged also. In freshwater ponds and puddles and ditches they are found, the N communae, that is nostoc communae, it grows in damp soil. Some species, they grow abundantly in paddy fields and they enrich the nitrogen content in the soil. Few species are symbiotically present in Anthoceros thalli, Cycas coralloid roots, Azolla and Lemna plants. There are few other species also which is a part of lichen. So the most common Indian species are Nostoc lynchia and Nostoc piscinale. Now we will talk about the plant body or the vegetative structure, in which we will take the topics of colony, the cell structure, cell wall, pigments present inside nostoc, the nucleoplasm and reserve food material. You can see the nostoc colonies here. So colonies, they may have twisted trichomes aggregated in a gelatinous matrix to form a ball-like globular colony, which ranges from greenish to bluish in color. The colony is externally bounded by a tough firm and pellicle membrane that provides a definite shape to it. The balls are olive green or bluish green in color, as I've told you. The size and shape of balls, they change with the age. They can be pinhead size or they can be size of a hen's egg. Example in case of nostoc pruniforme. The balls of nostoc amplissimum, they may reach up to the diameter of 30 centimeter. Shape of balls is mostly spherical or ellipsoidal. Sometimes it is irregular. Young colonies are solid, but at maturity they may become hollow or even break open into flat or lobed structures. So I'll show you the nostoc filament here. In this nostoc filament, you can see they are uniseriate and they are unbranched. They don't have branches. These filaments, they are embedded in the mucilage envelope. You can see the envelope around and they form a colony-like structure. Filaments can be curved, they can be contorted and they can be intertwined. The filaments, they possess terminal or intercalary heterocyst. Now I'll talk about the cell structure. Nostoc is a cyanophyceae member or a cyanobacteria which is prokaryotic in nature. It makes its own food, so mode of nutrition is autotrophic. So it is a prokaryotic type. The cell are without mitochondria, endoplasmic reticulum and Golgi apparatus. Vegetative cells are spherical or oval in shape, sometimes barrel shaped also. They are loosely joined with each other. They have a similar structure that of cyanophycean cells. In the previous video of cyanophyceae, I have explained the cyanophycean cells. So, because they are prokaryotic, they don't have a true nuclei. Each cell has a firm and rigid cell wall. The protoplasm is differentiated into outer colored chromoplasm and central hyaline centroplasm. So this is the cell wall structure. You can see the plasma membrane here. You can see the cell wall here. And this is the gelatinous coat you can see. Cell wall is made up of cellulose and it's surrounded by the protoplast. Now we'll talk about the pigments. The outer chromoplasm, it is colored due to the presence of pigments, chlorophyll A, keratin, xanthophin, and phycobilins. Phycobilins are of two types. Number one, C-phycocyanin, which give the blue color, and C-phycoerythrin. The photosynthetic pigments, they are located in sac like lamellae called the thylakoids. So you can see these are photosynthetic lamellae. Phycobilins are there here. You can see phycoerythrin red colored cells, and these white colored cells are bluish green colored cells are phycocyanin. So plastid are absent in case of nostoc. You can see the lipid droplets. This is the nucleoid, the circular DNA in the central part. Because true nucleus is absent, there is no nuclear membrane here. You can see the ribosomes, protein granules, photosynthetic lamellae, phycobilisomes, plasma membrane inside, outer is the cell wall, and there is a thick gelatinous coat here. So we'll talk about the nucleoplasm. The central centroplasm, it contains DNA and RNA. As there is no true nucleus or nucleoli, the reserve food material occurs in the form of cyanophycean starch here. And in nucleoplasm, as I've told you, there is no nucleus. Nucleus is absent. It is present in the central part. Lipid droplets, nucleoid, protein granules, and ribosome can be seen in the picture. Reserve food material. Reserve food material, I've already told you, it is cyanophycean starch. It is in the form of sugar, little bit of glycogen, and proteinaceous material called cyanophycean starch. So this is again a structure of soluble protein, soluble RNA, ribosomes, metabolites, and lipoprotein membrane. Now we'll talk about the reproduction. Sexual reproduction is absent in the case of nostoc, and you can see vegetative and asexual reproduction. There are few methods like colony fragmentation or hormogonia formation, akinets, arthrospores, and endospores. First, we'll take about the heterocyst. Heterocysts are specialized cells which are different from the vegetative cells. They can be terminal in position or they can be intercalary in position in the filament. Heterocysts are spherical, they are large, thick-walled, and they possess one or two polar nodules, depending upon their position. The terminal heterocyst has only one polar nodule, and the intercalary has two polar nodules adjoining the vegetative cell. These polar nodules, they have narrow pores through which the contents can be exchanged with the cells, vegetative cells. So asexual reproduction is done by heterocyst. In case of nostoc communae, the heterocyst functions as spore and they germinate to form a 2 to 4 cell structure which gets liberated after the rupture of heterocyst wall. You can see a heterocyst here covered by a thick wall. You can see the germination and growth of new individual from this structure. So heterocysts, they also perform other functions other than reproduction. Number one, they rejuvenate and germinate into new plant that you can see here. Secondly, they are the site of nitrogen fixation. Thirdly, they release some substances which promote growth of vegetative cells and formation of akinets. They may serve as storage organ also. So now we'll take the next structure. This is hormogonia. In case, this is the most common method in case of nostoc. The filament is broken down into many short length pieces and these are called as hormogonia. These fragments are formed due to development of heterocyst or death of any vegetative cell. You can see nostoc filament here. It is broken into two filaments and these two broken filaments will give rise to two new filaments, right? These hormogonia come out of the colony by piercing the colonial sheath. Each develops its own sheath and aggregate to form a new colony. The nostoc filaments within the colony, they may break up into small segments known as hormogonia, as I have already told you, and they can come from the region of a heterocyst or from a dead cell. The fragmentation can be either a cell in between a vegetative cell becomes dead, so that because of that fragmentation can occur, or a heterocyst also can be a reason because of fragmentation. Next, we'll take akinets. Akinets are also known as resting spores. You can see in this picture, this is a akinete. You can see heterocyst here. So these are known as resting spores. A vegetative cell enlarges and secretes a thick wall around it. Then it stores a large amount of food and function as a kinete. They are usually present adjacent to the heterocyst. They can be single or they can be in chains. They help plant during unfavorable conditions. When the conditions become favorable, these akinetes, they release their content through a pore that germinate into a new filament. So under unfavorable conditions, they are formed, akinetes are formed. Next is arthrospores. The resting akinetes are also known as arthrospores. You can see a akinete germination of akinete or arthrospore. These are endospores. In some species like Nostoc microscopium and Nostoc communae, heterocyst contents divide to produce endospores. These spores are released into new filament. You can see in the picture endospores. Now I'll talk about the importance of nostoc. Basically, nitrogen gas is present in the atmosphere but cannot be used directly by the higher plants. So, the biological organisms responsible for the conversion of atmospheric nitrogen into ammonia is blue-green algae and few bacteria, which helps in fixing the nitrogen in the soil. Nostoc enriches and fixes the nitrogen in the soil, and also it lives symbiotically in association with other plants. Like I have already told you, they are present in the coralloid roots of cycas and in the thallus of Anthoceros. Secondly, we can see the increase in grain yield of many rice varieties, as it has been observed that inoculation of rice fields with blue-green algae, there is increase in the yield. So nostoc is spread in the form of powdered flakes on the standing water in rice field at the rate of 7 to 10 kgs per hectare. So it enhances the nitrogen fixation and the yield of rice is more in that case. Third, balls of Nostoc communae, they are collected, boiled and consumed as the food by Chinese and the South Americans. Prepared food is also known as yoyucho. They are also used in a reclamation of Usar land, as all of you know that cyanobacteria or blue-green algae, they secrete some chemicals which counter the alkalinity of Usar soil. Next, we'll take some questions from Cyanophyceae and related to nostoc. These are few fill in the blanks questions, can be asked in viva of the practical or they can be asked in one-word questions in the theory paper also. So question number 1, blue-green algae such as dash and dash are the most common nitrogen fixing genera of the rice field. In the previous slide, we have talked about nostoc, right? So we can put blue-green algae such as nostoc and anabina. They are the most common nitrogen fixing genera of the rice field. Second question, Cyanobacteria resembles higher plants in having the pigment. The pigment name is chlorophyll A because in higher plants chlorophyll A is present, and in case of cyanobacteria chlorophyll A is present. Third question, reserve food material of bacteria is dash and that of cyanobacteria is dash. So reserve food material in bacteria is glycogen and that of cyanobacteria is cyanophycean starch. I have told you in the previous slide. Next is write the name of cyanobacterium known to produce toxins. So Oscillatoria, it secretes toxins. Now we will take few multiple choice questions. Photosynthetic pigment in cyanobacteria, they are located in A. chloroplast, B. thylakoid, C. stroma, D. chromoplast. So the answer is photosynthetic pigment in cyanobacteria, they are located in thylakoids. Question B. Some cyanobacteria, they live in symbiotic association with A. Cycas coralloid root, B. Anthoceros thallus, C. Azolla, or D. all of the above. The answer of this question is all of the above. Question number C. They do not possess flagella. What are the organisms which do not possess flagella? A. Bacteria, B. Cyanobacteria, C. Algae, D. Fungi. So bacteria possess flagella. Many algae have flagella and there are few fungi also which have flagella. Only cyanobacteria does not possess flagella. So the answer of this question C is cyanobacteria. I have written few more questions which are long questions. You can make notes of these questions for your theory exams. Like question number 3, what are heterocysts? How are they helpful in nitrogen fixation? It can come under 3 or 5 marks question. Question number 4, name the prokaryotic organism considered as the oldest of the living fossils. Question number 5, give the economic importance of cyanobacteria. Question number 6, describe the structure and methods of reproduction of nostoc. Question number 7, explain the role of blue-green algae in agriculture. There can be one-word questions also, like the other name of cyanobacteria is dash, or cyanophyceae and mixophyceae are same or different. So in Viva also these questions can be asked from the external examiner to you.